they're made of nucleotides. We talked about this last time, right? What are nucleotides? What are they made of? What's their structure?
Nucleic acid.
No, they make up nucleic acid, but they have a phosphate hooked to a 5-carbon
Sugar, yeah, and then hooked to one of the.
And then hooked to a nitrogenous base. And the sugar, for DNA, the sugar is called deoxyribose. And RNA is called ribose. Different by one oxygen, that's all. Is something falling from the...
Yeah.
What is it?
Something like asbestos right now.
Say it again?
Something dropped.
Well, I thought I felt a drop earlier. So
That's not good.
Is it water or what
I
is
don't know. I just saw something and I'm like, am I crazy? What was that?
White specks. Is it? We need to. I need to tell people.
I think it was because the door opening shut.
Yeah. Okay, but I want to make sure it's nothing. I mean, it couldn't be asbestos, I'm sure, because
No, we quit using that in '92. I was pulling it out in '92.
Yeah, right.
Yeah.
That wasn't good for you to do that. But anyway, so yeah. But if it's something that is happening, I need to report it because anything with the new buildings, they want to know anything, such as I think I mentioned to you that the urinals near the bedroom, the other building. One of them, I know that for you gals, it's not the same thing. Guys know about these, but anyway, for one of them, they're automatic flush, and I think I mentioned to you in class that one of them, it flushes and all comes rushing back out. You get it right back on you. So anyway, so they want to know these things. So if there's something happening, I did let me know how. Oh, let them know. Get rid of it. Okay.
Yeah.
But we'll go ahead. Anyway, but back to the nucleotides. Okay, so they're made of nucleotides, the DNA, but when they were trying to figure out the structure, they didn't know how the nucleotides fit together. Well, it turns out that the phosphates in sugar form what the actual helix itself. They form what they call the backbone of the molecule. Now, what they like to do, and it's a nice way to kind of think about it, is that you unravel the helix so that you make it a straight line, and then it looks, it'll look like this. It'll look like this, and so see, and then you see these things that makes it look like a ladder, and the things in the middle are the rungs of the ladder that you'd be stepping up on. But what Watson and Crick realized when they did all the different things they had to do to figure this out, and it wasn't an easy thing to figure out. It took a couple of years with a lot of different bits of information. They realized that what made up the actual, if it's a ladder, what you hold on to when you climb up, that's the sugar and the phosphates. So the way I would usually draw it is like this. I'll say phosphate, sugar, phosphate, sugar, phosphate, sugar, like this. And then on the other rung, the other ladder, I should say, the other side of that, the other handle of the ladder, you have phosphate, sugar, phosphate, sugar. For you guys who are real purists about this, these two different strands are said to be anti-parallel, which I don't really want to talk about. One's running up and one's running down. It's not important for our purposes, but anyway. So, here's what Watson and Crick realized, or figured out, I should say, is that the backbone were the sugar phosphates, and what forms what would be the rungs of the ladder are the nitrogenous bases, these so-called nitrogenous bases. Remember, there are 4 different nitrogenous bases. And what are they? Well, we don't only have to have the letters. I don't want you to worry about that.
A, T, G, and C.
Okay, so those are the 4 bases. Now, here was the critical thing that they discovered. Yes, those are the bases. They make up the rungs of the ladder if you're stepping up the ladder and walking up. But if there's a certain base here, let's just put them up here. Let's just say that we have, I'll do it in this color. Let's just say you have an A here, and a G here, and a C here, and a T here.
That's going to be a U.
If you have a certain set of bases on one strand, there has to be, no choice, there has to be only certain ones who can fit with it.
Mm-hmm.
Okay, and this is what they call the base pairing rules. Now nowadays people call them the spell that right, Watson-Crick base pairing rules. They didn't call it this. But most of the time I'll use the word complementary base pairing. What they showed was that if you have an A on one strand, the only possible base that can pair with it on the other strand is a T. So A pairs with
this is
T.
DNA, not R.
And of course, if you have a T, only an A could be on the other one. And C pairs with G.
G and B and C.
So as soon as you know what one set of bases are, you immediately know what the other has to be. So if A is on this strand, the opposite strand or the complementary strand would be the right word, would be a C. Oh, sorry, a T here. Sorry, I'm making a big mess. I have to have a T here, then a C here, then a G here, and A here. And they actually form ionic bonds. There's special kind of ionic bonds called hydrogen bonds. I'm not going to really worry about it, but they're actually ionic bonds. But that's what holds these things together in a nice stable way, with all these millions and millions of. I mean, I showed you what 4 nucleotides. And by the way, just to make it so you see it, let's just put a little. I should have the color there. I might have the color. That works. This would be one nucleotide. That's one nucleotide. So so if you have nucleotide, nucleotide. Well, how long is this going to be? I showed you 4 bases. It may be a million bases long. I mean, we're talking about a long strand here. I'm showing you one tiny little piece, right? So, and so let's just also I'll label this and say this is one strand of a double strand. Here's one strand. The one on this side would be the complementary strand. The one you said doesn't. I'll be right there. It's a complementary strand because of the base pairing rules. Only once you have one strand, the other strand has to be a certain way. Otherwise, it doesn't fit together. Sounds like you.
I was wondering, you have the sugars connected
With the sugar, phosphate, yeah.
Yeah, but you have only the sugars connected to the bases. Could the phosphate connect to the bases? Only the sugars?
Just a sugar. Okay, so if you just think of this would be the double strand, and of course it's a long thing and it does form a helix, so it's a double stranded helix. I've opened it out flat so we can see it much more easily. But this is the critical thing. These base pairing rules are critical because, first of all, remember I put it up here. The model showed, 1, how DNA could be the genetic material and how DNA could be used to tell the cell how to make proteins. We're going to come back to that in just a second, but this is basically what we're building up to, is that from the model itself, you can certainly start to say, oh my gosh, because of this base pairing rules, you can only hook things in a certain way. It gives me some ideas of how maybe this could be the genetic material. Let's talk about that for a second. But before I do it, I just want to make sure you understand this idea of the base pairing rules. And they use the idea of a template. Use a template if you like. Template. And you use templates for. As a kid, you like to do nice. You get a little thing with letters, and you can. But they've got the cutouts, and you put your little pencil in there and you got the letters, So that would be a template. But I mean, you use templates in all kinds of ways to do things. So the idea is this is, if you have one strand, that is the template to tell how to make the other strand, and you can only make it in a certain way. So let me tell you that in terms of the genetic machinery. the fact that DNA is the genetic information in two different ways. One is it's the information that's passed from generation to generation to generation. So it's in the seeds and the sperm and the eggs, and then that's passed on to the next generation, et cetera, et cetera, et cetera. But even cell to cell, when a cell divides, the information of the DNA in your nucleus is duplicated exactly so that the two new cells, and remember I use the term daughter cell, that's a term we use when cells divide, the two daughter cells have the exact duplicate information as the original cell had. Now, how could that be? That was a big question. That was a big question. How could, before they knew the structure of DNA, they were pretty sure that it was being duplicated pretty exactly. But how could it be duplicated so exactly? There's got to be some technique. Well, then once they saw the structure, they go, whoa, hey, here's the deal. To duplicate DNA, and the proper word is replicate, so I'll use the word replicate, but I'll put parentheses duplicate. To replicate DNA, okay, so this is what we want to do. If the cell's going to divide, you're going to have to make totally new information, totally new copies of all the DNA so it can be passed to each daughter cell. But we want to duplicate it exactly. So to duplicate or replicate DNA, and I'm going to oversimplify the way it actually works. In reality, it's a little more complicated, but effectively, this is what happens. Okay, separate the strands. The two strands, right? This is the double helix. Separate the two strands. I'm writing the two there. Separate the two strands. And use each strand as a template,
For matching.
to make the opposite strand. In fact, I say opposite strand, I would actually, instead of writing the word opposite, complementary strand, because they are complementary. You can say opposite strand, I don't care, it's okay. I'm going to use the word complementary. By the way, this word complement, I said this in the lab, a couple of you were in the lab this morning. Notice there's an E in the middle of that word. It's not like the one with an I in the middle. If you had an I in the middle, that would be, if you said to someone, wow, you have a really nice double-stranded helix there. That was paying somebody a compliment, saying something nice about it. That's not this word.
That's
This
complete.
word compliment means they have some relationship to each other. And in this case, the complementary relationship is this Watson and Crick-based pairing rules, that one strand, the first strand and the second strand aren't identical, they're complementary. Where you have an A in one strand, there's a T in the other. You have a C in one, there's a G in the other.
They complete each other, yeah.
Complementary to each other. At any rate, the point is that if you do this, let's do it. Let's just do it. We can do it right here, real quickly. So we'll separate these two strands. I just made simple strands. So let's just do it. We'll take here's one of the strands. Okay, and we had what? We had A, G, C, and T. Okay, and I'm separating it. So here's the other strand. This was S, E, S, phosphate, sugar, Okay, and then we had T, C,
G, A.
and A. Okay, so those are the two original strands. We've separated them. Now we'll use this left-hand strand right here as the template to make a new right-hand strand. And as the strand's being made, here we'll do a different color here. As the strand's being made, the only nucleotide that can fit in place is the one that has a T on it, or the first one here. And here's the next nucleotide that can fit into place. The only one that can fit has a C right here. And the next nucleotide that can fit into place
Has a G.
has a G here. The next one that this was possible fit into place has an A here. So when you look at this is identical to the original strand. And of course, you do the same thing on the other one, and you get another one that's exactly identical to the original. You know, so I won't necessarily draw it, but I mean you can do it. But the point is that you use each strand as a template to make the complement, and you end up with two new double helices that are identical to the original.
They're just a draft.
Yeah, it keeps opening. You want the door open or closed? I mean.
No, I just thought that might have had something to do with
Yeah,
the stuff following.
You want to close or open? Oh darn! It's getting hot flashes or something. Go in close. I'll do whatever you need. So two new double helices. Oh god, that's plural helices. Okay. Two new double helices that are identical to the original. I don't know if I needed to write the wording helix and helices here. I could have just said two new bits of pieces of DNA that are identical to the original, whatever. But the point I'm trying to make is it was the model that showed how it could possibly be done. So then you could ask that question, hey, is this the way it works? Then you could go ahead and do experiments that prove that yes, that is actually the way it is done. And that's what they did. You know, and I don't think these experiments were easy and quick. It took a long time. Now they're incredibly sophisticated. We're talking back in the 1950s and early 60s. Now they're very sophisticated with what they can do with DNA. And the interesting thing is you read about this in the paper every so often. They used to think that DNA was really fragile. And it turns out because of all the ionic bonds that are formed. I mean, you know, sometimes what we do, we put little dots in here that indicates the bonds, you know. But you don't have to put that in, I don't care. But the fact that you have these long strands of DNA and they're held together by, thousands and thousands of these ionic bonds, it turns out that DNA is actually very stable. Now, it's not stable against things like, high temperatures or, change the, pH or salt content.
Like gold.
But in terms of just getting bits of tissue that have been around for a while, not fossilized, but if you get tissue, you can extract the DNA from it. And so they've actually been able to extract not all of it, but like from a mammoth. When was the last time there was a woolly mammoth? About 10,000 years ago or something like that.
Yeah.
But they've been able to find, some of these things that have been
10,000
frozen in the
years.
ice in the permafrost or whatever, and they can take tissue samples. They can't. It's hard to get the entire perfect genome. That's pretty difficult. But they can get stretches of DNA, so they can act. And if they can get the DNA out of something like that,
You can make it.
they can then separate the strands and have it. They have mechanisms now to get it to make many, many, many copies. They're actually hoping that maybe they could remake woolly mammoths, by getting some of the DNA from an original mammoth and then trying to put it into like
Elephant
eggs,
Mhm.
and then try to put exchange some of those and maybe try to get a mammoth and have it being raised by an elephant. Who knows? It's possible.
It'll be way bigger. God.
Yeah, well, I don't know. I mean, it'd be interesting if it
Yeah,
happened.
yeah,
They're actually
I'd
thinking
be
of it. They're
stoked.
trying. I think they're trying it. So it's a cool idea. But they've even been able to find things that are even more thousands of years old than 10,000. And the DNA is
Jurassic Park.
intact. Now, don't expect it to be fully intact. I mean, I'm not saying the entire genome.
Yeah, it is.
I mean, bits of it. So it's kind of wild stuff. Okay, but anyway, so back to this. So the first question would be, how could DNA be the genetic material, meaning that it can be duplicated, replicated exactly. I'll use replicated.
And it's also directional, right? Like it has order.
Well, yeah, it does, but we're not saying we're going to worry about it.
Okay.
Okay, it can be replicated exactly and passed from generation to generation. Now, next lecture, we are going to talk about generation. Right first.
Okay.
We're going to talk about mitosis, cell nuclear division and cell division. So I'm talking about it can be replicated exactly and passed from generation to generation. But you can think about it, as I said, in terms of cells dividing and passing exact copies to each new cell, or passing this information from generation to generation. But the point is that you want it to be passed exactly. Now, mutations can occur, and we'll talk about a mutation in just a second. But most of the time, it is passed very much exactly. And the weird thing is, there are even repair enzymes. Enzymes that can, remember, enzymes, they do all the work, right? DNA is not doing any work. DNA is informational, but enzymes are doing the work. They're the ones that are making all this work. There are a whole bunch of enzymes involved in replication of DNA. But there are even repair enzymes. So if a mistake is made, the enzymes are copying and trying to make a new copy, and they make some mistakes. It's not fitting together correctly because these only fit when they're the right complementary base pairs. If there's a mistake made and they're not fitting together, one side will get cut out, and the new stuff will get cut out, and it'll be replaced with exactly. So mistakes are not common. Mistakes are rare. So it's kind of wild. They're even repair enzymes. They call them proofreading enzymes. That's if someone's typing and you read over, try to catch it. Well, now we have those things on spell check and grammar checks. No, I don't like most of their grammar checks, and my spell check doesn't know most of the words I use because they're science words.
Right. No.
So I have to educate my
Add to dictionary.
Yes.
I do that all the time.
Exactly.
Yeah.
But the point I'm making is that when you're making corrections, you read over what you did, you make corrections. That's called proofreading. So they use that same word when they're talking about making new DNA copies. Proofreading to make sure it's correct, and if it's not correct, cut out the bad stuff and replace it with the good stuff. So it's kind of wild how all this works. All right. So that was. So I talked about the replication of DNA. I'm not going quite in order here, but I needed to talk about the replication of the DNA to answer this question in terms of how the model actually shows how it could happen. I should talk about mutations. Then we'll talk about the central dogma of DNA, which we'll get to this idea how that information can be used to make proteins. But so let's talk about what is the deal. Let's get some words here that we need. We haven't used the word chromosome. I was going to wait to use the word chromosome until I talk about mitosis, cell division, but I think I could use it. We have a lot of DNA in our cells, and by the way, only roughly 2% of it is used to have the information to make protein. So we have a whole bunch of extra DNA, and for a long time, people, in fact, Crick was the first one who used the term. They called that junk DNA. They didn't know what it was for. And some of it probably is junk in the sense that it's just leftover stuff from millions of years of evolution. It's not doing anything. But probably most of it isn't junk, is my guess. Most of it's probably helping to regulate the way the other stuff is
Source
being
of
used.
archetype.
So it's
or
hard
memory.
to know exactly, but whatever. Only roughly 2% of the DNA actually has information to make proteins. But that's really what DNA has. So let's say DNA has, and I've told you this before. This isn't anything new to you guys. Has the information to make proteins. Remember that the DNA doesn't do any work, of course. It just has the information. But it has the information to make proteins. And we use the word gene. We use the word gene. A gene is the segment of DNA, portion of DNA. I don't know, segment, maybe it's the portion, length, nucleotides, whatever. The portion of DNA with the information to make one protein. So it's the gene. Now this has changed slightly over the years. Originally, the people who came up with this idea was back in the 1930s. One gene was the information to make one enzyme, and I think I might have mentioned this last time. Then they realized, no, it's got to be more general than that because a lot of proteins aren't enzymes, so one gene for one protein. It's also the case that some proteins are made of more than one polypeptides. Hemoglobin, for example, is 4 polypeptides work together, fit together. So now a lot of people will put one gene, one polypeptide. There are other variants on it. Whatever. This is so let's write the way they usually write it. They write one gene, one protein. And if you want to say one polypeptide or whatever, they're even more sophisticated little variants. We don't need all those sophisticated variants. But if I ask you what a gene is, I just want you to say, hey, it's DNA information to tell how to make one protein.
Yeah.
See, that's it. That's easy. What's the word chromosome? Chromosome. Is that quacking? Is that what? That's. I like that. My daughter would probably want that if I know where you got that. Anyway, one gene, one protein. Okay, let's use these other words. So how much other words that I should use right now, just to get it? If I use the word chromatin, or some people pronounce it chromatin, that's okay. Chromatin is all the genetic information, all the DNA in your nucleus. I'm gonna write that word down. Chromatin. That's what most people pronounce. But let's say you can say chromatin. Chromatin is all the DNA in the nucleus. To be technical, it actually includes. I said last time, when what was the nature of DNA? I asked that question, and that was a question that was took a long time to answer. For 50 to 100 years, people were saying, what is the genetic material? Is it DNA or protein? And what's in the nucleus is roughly half DNA and half protein. So they didn't know for a long time, but there is a lot of protein in there. So it's all the DNA in the nucleus and the protein, if you want to be technical. Most of the time, I won't talk about the protein in the nucleus. What is the protein in the nucleus? There are all kinds. So there are all kinds of enzymes, for example, and these enzymes are necessary to get everything to function properly. There are also some proteins that are packaging kinds of things. The DNA is packaged around some of the proteins. They're very important proteins. You don't really talk about it much unless you take a cell biology class. They're called histones or histone proteins. So there's all kinds of proteins in the nucleus. So most of the time when we use the word chromatin or chromatin, we're thinking just DNA. But, if you want to really be technical, you would be thinking proteins too. The genome would refer to all the actual genes that you make up your DNA. It'd be all genes in your DNA. So, and the thing is, they sequenced the entire human genome a few years ago, 3 or 4 years ago. And I mentioned to a couple of you, it was kind of a shock. Humans, I am glad I'm a human, but we think we're so amazing. We are in many ways, yes, but we're kind of deficient in other ways too. I don't want to be speciesist about it and pretend that we're so great. I'm glad I'm a human. I definitely am. But we've gotten a lot of shocks over the years, and one of the shocks we got was when they sequenced the entire human genome. Because, 5 years ago, if you'd asked me how many total genes do we have, what's a gene? A gene is the information to make one protein, right? If you'd ask me how many total genes do we have in our cells, I'd say, well, I don't know, but, the estimate is anywhere from, 10,000 to 100,000, but probably, I'll just come up to the middle thing and I'll say 50,000. But a lot of people thought it was probably closer to 100,000. So they sequenced the entire genome. And this is the shock. It turned out we only have like 29,000 genes. They didn't think that was enough to make us so special. I mean, fruit flies have 20 some odd thousand genes. I can't remember how many. The little tiny bacterium, that you can't even see. You need a microscope to see it, but the little bacterium called E. coli in our gut, they have 2,500 genes. So, gosh, we're just barely more than 10 times. We have more than 10 times the number of genes of this little quote lowly bacterium. Just didn't seem enough. Well, it obviously is enough. Whatever, it's enough. But
Yes,
we
the
just
bees
assumed.
comply
I
with
mean, I would have
physics.
said, I'm surprised. I would have thought we had 50,000 to 100,000 genes. All right, we only have 29,000. We're okay. We're still okay. But when you say the entire genome, that's what you're talking about. This finding out all the actual sequence, when they say sequence of the genes, they mean take the first gene. How many nucleotides are they, and what order are they? A, G, G, C, G, A, C. That's what they mean. They have actually sequenced this. But how big is a typical protein? Now, what we're going to see is that the gene is the information to tell how to make a protein, and we're going to see that for each amino acid you need 3 nucleotides. We're going to get to this in a few minutes. But how big is a typical protein? Well, they vary so much. So again, if you want me to give you an average figure, I'd say, an average protein may have 150 to 200 amino acids in it. That's how long it, not where they fold and whatever shape they are. But there are proteins with thousands and thousands of amino acids, some short ones. But on the average, you could just roughly say, okay, hey, each protein may be 150, 200 genes. I mean amino acids. So, how many nucleotides would make up that gene? Say 500, 600, something like that. So, if you just say, okay, you figure an average gene may be, say, 600 nucleotides long, and you have 29,000 of these, that's a lot of nucleotides, that they had to sequence. And by the way, that's only like 2% of all the genetic material we have. So, well, where are we going from this? No, right, you don't need to worry about it. I'm just talking, but it's just kind of interesting stuff. Okay, so let's talk about a gene determines a protein, right? It has the information to make a protein. The way, well, I better tell you, before you talk about mutations, maybe I better talk about the simple dogma after all. I'm going to start here, and then talk about number 2. And we're going to talk about all these, number 3 too. I mean, that's 3. I got to talk about all these things. I can't just jump into from one to the other. I got to go in some kind of order that makes sense. So let's do this. Let's talk about the central dogma. In fact, this is what you guys are doing in lab this week anyway. Central dogma of molecular biology. That was the term used by Francis Crick. And people got all upset because there's no dogma in science. He's right, there's no dogma in science. And he was being funny. It was a joke. He didn't mean it seriously. But the central dogma, and that's why I put it in quotation marks, central dogma of molecular biology. And you'll see this written as DNA, an arrow, RNA, with an arrow, protein. They'll put the words from DNA to RNA, put transcription.
And translation.
And translation for RNA to protein.
Mm-hmm. A transcript is just a copy, some kind of a copy.
So what we're saying is, okay, DNA, you make a copy of the DNA in the form of RNA, and somehow that RNA is going to be used to tell the cell how to make the protein. So in other words, we're going to translate, I should say, that information in the form of nucleotides into the form of amino acids. So transcription means making a copy of the DNA, let's just say a gene. We'll just make it a gene. Copy of a gene, because a gene is DNA, right?
Mm-hmm.
Transcription means making a copy of a gene in the form of RNA. And it's a particular kind of RNA as far as our name. We call it messenger RNA. So I'm going to put that in and I'm going to get it explain all this in a second. Okay, so transcription then means making a copy of a gene in the form of messenger RNA. Now that mRNA seems, well, I'll write that down in a minute. Let me answer your question.
I guess I'm a little confused.
Okay.
It's a transcript, a DNA is a gene, but then you say gene is a protein.
No, I didn't say gene is a protein. A gene is the DNA information to make a protein. So, but when they say one gene, one protein, they're not saying they're the same. They're saying, hey, one gene is the information to make one protein. Okay. Yeah, make sure that's careful because they're totally different things, right? A gene is nucleotide bases. Protein is amino acids, and those are so totally different. No relationship. Okay, but now I said something about RNA. We need to talk about RNA real quickly. See, you can't just suddenly jump into everything. You have to do it step by step. So let me talk a little bit about RNA. So there are three types of RNA. First of all, what's RNA made of?
It's made of
It's similar.
the phosphate and the
So RNA made of nucleotides, I should say, but slightly different. So in this case, an RNA nucleotide
has uracil instead of what you call it,
thymine.
thymine, yeah.
So an RNA nucleotide is made of so one, phosphate, that's the same. Two, sugar. It's a slightly different sugar.
5 carbon.
It's still 5 carbon.
Yeah.
It's called ribose.
Yeah, right. Yeah, and the other one's
DNA,
deox.
in DNA, it's called deoxyribose. The only difference is missing one oxygen. And I think I said this last time, or maybe it was you guys, but it doesn't make a big deal to us. But it is a big deal to DNA and RNA. It is a big deal. But to us, it's not a big deal. It's ribose. I'm going to put parentheses as opposed to deoxyribose. And you don't need to, I'm not really worried about this. Yeah, if you just notice the 5 carbon sugar, ribose, deoxyribose, and DNA.
Which means it's removing one oxygen.
One oxygen. Just one oxygen less. Nothing for us to worry about. Okay, so still made the same phosphate, sugar, almost the same sugar, not quite, and then 4 nitrogenous bases. And they are A, U, C, and G. Okay, A, U, C, and G. So there's where DNA had a T, thymine, RNA has a U. Yeah. That's 5 carbon sugar. That would, yeah. What did you think it said?
I wonder if it meant 5 carbon or if it meant
It's a 5 carbon sugar. As opposed to glucose, which is 6 carbons. These are 5 carbons. And the base pairing rules now, complementary base pairing between DNA and RNA would be almost the same as we had, but it could be A going with U and C going with G. So again, where you expected T is U, that's all. One other thing about RNA and DNA, I want to make a little comparison, then we're going to I'm not done yet. We're going to keep on going. I didn't tell you about translation, but I'm going to. Let's just say a couple things about DNA and RNA. Just comparing it. DNA versus RNA. Okay, so DNA always double-stranded in nature. Double-stranded in nature. Whereas RNA is always single-stranded. In nature. Now, there's an absolute there. Always. God, and I hate to put those absolutes in case there are any exceptions. And there are exceptions. And in molecular biology, when you come up with a rule, and if there happen to be any exceptions, usually it occurs in some viruses. So nothing you need to worry about, but just for your knowledge, you don't have to have it anywhere in your brain, but there are some single-stranded DNA viruses, and there are some double-stranded RNA viruses. So nothing you need to worry about, but just so you know, there are. If there's an exception in molecular biology, it's usually some virus. Yeah.
The in nature part, do you mean that in nature to be double standard or when it's
No, I mean this is where it occurs normally, as opposed to in a test tube. You can take double-stranded DNA and you can heat it up and get it to separate. So and that's what they do in all kinds of things they do, for genetic engineering and all that. So when I say in nature, that's the way it naturally occurs, naturally occurring, as opposed to what we can do in a test tube. Now science can do all kinds of things with it, including making all kinds of alterations. Okay, so just for comparison purposes, DNA is always double-stranded, RNA is always single-stranded. DNA, the sugar is deoxyribose, and in RNA, the sugar is ribose. And the only difference is deoxy means one fewer. They're both 5 carbon. It's just that the only difference is one less oxygen on the deoxyribose.
It's not an OH, it's just a H.
As I said, it doesn't matter to us. But the way they often write the word, I mean, instead of writing out the whole thing, deoxyribose, what they'll do is they'll put a small d and they'll put ribose like this. So if you happen to see a small d in front of ribose, then they're talking about the sugar for DNA. Okay, that's all. Deoxyribose, but that's just a short way of writing it. And then the other thing that makes it one different from the other is the bases. Bases are A, T, C, and G, and then RNA is A, U, C, and G. All right. Okay so far?
Mm-hmm.
This is all wild stuff, actually, when you think about it. This stuff was all learned in the last half of last century. And in the 70s and 80s of last century, that's actually the last millennium, isn't it? Sorry, the previous millennium. The last couple of decades of the previous millennium. That's how long ago it was. Makes it sound like a long time ago, doesn't it? That's when they, started the whole thing of this genetic engineering or recombinant DNA technology. And now they use this, the genetic engineering and doing all kinds of things. It's just incredible kinds of things they can do. And as I said, you really go back to the Watson and Crick model that allowed all this stuff to be discovered because you could ask the right questions. You could ask the right questions, then you could test it and see what the answer was, and you could go on from there. But before you could ask the right questions, how would you know what to do? So it's kind of amazing. All right, I haven't gotten to this part yet, but I'm still there. I'm getting there. So one other thing. Let's do one other thing. Transcription means making a copy of a gene in the form of messenger RNA. So let me say something about. Oh yes, go ahead. I'm sorry.
What is translation?
I'm going to get there. But it means basically what it means. I'm going to write it down, but I want to say something first before I get it. I'm getting there. So leave some space here. Okay. But What it means is taking the information in the form of nucleotide bases and using it to put amino acids together. So you're changing the information from nucleotide bases to amino acids to make a protein. I'll write that now. What the heck? Let's just write it now. Seems like a good idea. I was going to explain more about transcription, which I'm going to do. So, translation. Using messenger RNA information to put amino acids together to make a protein. And just to say it again, 3,000 times, but the proteins are the worker molecules, but each protein is highly specific. And to make that whatever that protein is, you have to have some specific amino acid as the first one, and then the next one in place, the next one in place. They have to be in perfect order, and of course they start folding into whatever form they're going to make. But it doesn't. It's not random. I mean, a protein is not just a random assortment of amino acids. They're put in a particular order, and you can use the idea of alphabet and putting letters together to make words. You put the letters together. Q, J, X, C, and you go, it's not a word. You put Q, U, I, T, and you go, yeah, it's a word. So there's certain combinations that work and certain ones that don't. It's just that words are short compared to proteins or genes are long. Yeah.
I'm not understanding what messenger RNA is.
No, I'm getting there. That was the next thing I was going to say before I wrote that. But since you asked this question first, I said I'll do that first. So I am going to do that right now. So, okay, so let's talk about a little bit about RNA, so then we can talk about translation. RNA. I already told you a little bit about it, all the single strand and all that. But there are three types of RNA. Three types of RNA. One, we call it small m, big RNA, which means messenger RNA. Okay. But before I actually write down what it is, let me just tell you something. When they first came up with this idea, the so-called central dogma, they're going, okay, this all makes sense and everything, but there's a problem. I shouldn't say this before they knew about this middle part here. They knew that DNA had the information to make a protein, but they go, wait a minute. DNA is always in the nucleus. Proteins are always made out in the cytoplasm. So there needs to be some way to get the information from the nucleus to the cytoplasm. And someone came up with the idea, if you know the right question to ask, he said, most likely, what could read DNA? How could you copy DNA? Not in the form of DNA, but something similar, RNA. So they said probably there is some RNA molecule that takes that information from the nucleus and takes it into the cytoplasm. And so then they did all kinds of experiments and they proved, yes, there is such a molecule. We now call it messenger RNA. And we usually just write little m, big RNA like that. It means messenger RNA, but this is messenger RNA. This is a transcript of a gene. What's a gene now? DNA information to tell how to make
one protein.
One protein. Okay, so this is a transcript, copy of a gene that goes to the cytoplasm and hooks up with the little protein synthesis factory.
And it fills in the blanks.
Ribosomes. That goes to the cytoplasm and hooks up with a ribosome. So a protein can be made. I'll just say made, but synthesized is the way I like it. Say it. So we'll come back to ribosomes in just a minute, because those are the protein synthesis factors. We're not going to get into huge amounts of detail. We don't have time, but it's amazing the way it works. Okay, the second kind of RNA is called transfer RNA. So little t, big RNA means transfer. These are small little molecules. I mean, relatively speaking, they're only like 50 amino acids long or something. But these are well, let's before I write anything down, let's think about it. RNA is what kind of information? Nucleotide bases. Protein is made of what? Amino acids. There's no relationship between the two. There's no relationship between nucleotides and amino acids. And yet, if the information in DNA copied into the form of messenger RNA is going to be used to make a protein, we have to have some kind of molecule that can combine those two kinds of information. And it turns out it's the transfer RNA molecules. They used to call them the adapter molecules. We don't use that term so much anymore, but it's kind of neat. But the adapter molecules, or the transfer RNA molecules, these guys, what these do, these are relatively small RNA molecules. And by the way, they're bent into a shape, a funny shape. If I drew a picture of a transfer RNA, they tend to look sort of like this. So this is like a kind of a transfer RNA molecule, something like this, kind of a cloverleaf form they call it, and where you have things together like this, there's actually some bonding, some complementary base pair bonding. It's not a double helix. Notice it's not even a helix at all. It's like a single strand, but it does kind of fold in on itself, and there are parts of it, several places where it actually bonds together. But the tricky part about it is there's a part down here, that can recognize the signal, recognizes the
mRNA.
Recognizes
mRNA.
I'm going to use the word codon, which I haven't given you yet, but I'm going to.
Oh, right.
The codon signal on messenger RNA. Okay. So that's what this is right here. It recognizes the codon signal on messenger RNA, the codon information. I said signal, I don't know, signal's not the right word. I should say codon information. I couldn't even say codon, but I haven't go to a codon engine. I'm going to say the codon information.
We found out
the codon information on messenger RNA, and by the way, it's called the anticodon. Can you read this in the back? In red, can you read it? It's the anticodon. And
it comes up on the screen.
Up here, I have my word letters in the way. Up here, it's carrying a particular amino acid. So it's the adapter in the sense that it can recognize the information on the messenger RNA. Let me see if I can get this to. It's called the anticodon, and it recognizes the codon info on the messenger RNA. I haven't told you what a codon is, but I'm going to. But it also carries an amino acid. So it's what
codon
can get the two types of information connected. Nucleotide information with amino acid information. So these are small RNA molecules. There's 61 different ones. Each one can recognize. going to tell you the codon in a minute, so just write it down now. Recognize a codon of messenger RNA plus it carries an amino acid. And I put AA up there for amino acid.
And that's can connect.
Say it a little louder.
And that's to make the protein amino acid.
Yeah, so that's one amino acid that's going to, be there, and then there's going to be another one with another amino acid. They're going to get hooked together. It's all going to happen on the ribosome, which I'm going to tell you about also. I do want to stop and tell you a little story. This so I don't know, sort of an interesting story. I think kind of a sad story. It's kind of pathetic, actually. But I was on sabbatical took a sabbatical at Cambridge University in England, and over there's some incredible numbers of Nobel Prize winners and all that. But anyway, and I was at this research lab, virus research lab, across the street from a big research center, and now England has all this class structure. I'm not a Californian. I'm not used to class structure. The only class structure we have in this country is the wealthy and everybody else. 1% and then everybody else, But so other than that, we don't really have any class structure. So I go over to England and there's all this class consciousness and stuff. And it's just kind of foreign to me. But at any rate, as part of this research group, we were allowed to join a kind of a health club that was associated with all this. And the big MRC, Medical Research Council, that's probably gotten more Nobel Prizes per square person than anywhere else. And it's this incredible research institution, and it's just right across the street from where we are, and we were kind of associated with it. And so they have this health club that you could join. So and so when I was in the lab, they said, "Oh, you should join the health club." I said, "Great, sure, why not? You can eat there. They've got sort of squash courts. I played squash once. It was a big deal, but whatever. They do all these things, and so I remember one time I was talking to a guy he was a very famous historian, and I said something about the health club. You remember that? Yeah. Wow, that's pretty neat. I go, oh, really? I didn't know being a member of this health club was such a big deal.
Prestigious, yeah.
But anyway, whatever. It was if you had to be a science person to be a member of this health club. So anyway, the whole lab, went over, and the guy who was the head of the lab we all got, they call them hamburgers. If you're an American, you kind of look at their hamburgers and kind of scoff a little bit, but it's okay. They tried. So we all went over. We had hamburgers or cheeseburgers and whatever else. Stuff like that. So we go back to the table and start eating. But I noticed the guy who was the head of the lab was talking to some guy, and to be frank, it looked like he lived on 2nd Street in Eureka. I mean, he was this old, what I would have called an old wino, and he's talking to this guy, and I'm thinking, who could this guy be who's a member of this health club? Because he has to be a member of one of these big research groups in order to be a member of this club, because otherwise he wouldn't have been in there. So when he came, the head of our lab came back to the table, so I said to him, out of curiosity, who was that guy you were talking to? He says, it's kind of a sad case. He said, it's John Smith. Now, I know it sounds like a fake name, but that's his real name. John Smith. He's one of the co-discoverers of transfer RNA. He's one of the guys, there are two, there's an American and English, him, John Smith, who were just the first ones who discovered and proved that transfer RNA actually existed. He said, but unfortunately, he's become an alcoholic, and he comes in the morning and reads a few papers, maybe messes around the lab, and then starts drinking in the afternoon, and that's the end of the day for him, Goes on from there. So, it's sort of sad. It really was kind of sad, but I'm going, God, there's the guy who discovered transfer RNA. I mean, that's pretty impressive. So anyway, whatever. So that's my little sad story. One last thing, and we're going to come back to all this. Notice I said there's 61 different ones. Number 3, here, let's do number 3 here. Number 3, the third kind of RNA is called little r, ribosomal RNA. So So the little r stands for ribosomal. Ribosomal RNA. It turns out that ribosomes, I mentioned those to you already, and I'll say a little more about them. Ribosomes are the protein synthesis factory. Okay, they're made of two things. They're made of ribosomal proteins, ribosomal RNA. That's the last you need to worry about ribosomal RNA. We're not going to talk about it again. It just helps. It's part of the structure of ribosomes. Ribosomal RNA combines with. You can see this ribosomal proteins. To make ribosomes. I'll just say a little bit about ribosomes right now, and then we're going to come back and answer words like what that word codon mean and all that stuff. We talk about the anti-codon and stuff. So we talked about eukaryotic cells. And what's the big difference between eukaryotic and prokaryotic? Eukaryotic have a nucleus and other membrane-bound organelles. The nucleus is a membrane-bound organelle, so are mitochondria, they're membrane-bound organelles, endoplasmic reticulum. Ribosomes are a kind of organelle, but they're not membrane-bound. Okay, they are not membrane-bound. Bacteria have them, our cells have them, everybody has them, okay? But they're the protein synthesis factory, and they're made of combination of ribosomal proteins and ribosomal RNA. No big deal for our purposes as far as their composition, it's just that they are the synthesis. In fact, I should write that up. I'll write that up, that ribosomes are the protein synthesis factory. And in a given cell of yours, if you just took one of your cells, these guys are tiny. You have maybe 100,000 little ribosomes in any one cell of yours or something like that. I mean, they're tiny little things. And of course, they're sitting there ready to do their thing when they need to, which is the protein synthesis factory. So a ribosome is a non-membrane bound organelle. So everybody has them. Every cell type has them. They're made of right there. The function we have said is the protein synthesis factory. And I'll put factory in quotation marks, but you've all seen places like bottling Coca-Cola or something like that, and the bottle comes through, the stuff goes in, and the top goes on, and everything goes back. That's the way these things work. And by the way, they're made of two parts. There's two subunits. It would look like this. And what happens is, so this would be the ribosome in black. The messenger RNA feeds through it like this. Okay, so in black is a ribosome. And the way I like to do it, the analogy I like to use, is a tape recorder. Now, everyone uses digital stuff now. So some of you remember tape recorders, maybe.
Yep.
And the tape, is magnetized and it goes through the tape recorder and the tape head and recognize the magnetic changes and transfer it into electrical signals, which will then make sound and you get to listen to music and stuff. Well, in a similar way, the messenger RNA is going through, this is going through, and in the process, transfer RNAs come down. So we need to talk about the codons and the anti-codons. I'm going to give you a hint about how it all works in the final analysis without going into the detail, and without expecting you to go into the detail, because it's complicated. But it's kind of wild. So the codon. So they discovered transfer RNA, and then they said, so transfer RNA, is sort of that adapter molecule that can take the genetic information in the form of nucleotide bases and somehow translate it into the making of proteins. There must be some code. There must be some code that relates signals on the messenger RNA or bases on the messenger RNA to amino acids, and that's the so-called genetic code. Well, they discovered it. And actually this one guy discovered the first code letter, and I mean code signal, and he presented it at a meeting. And, oh my God! All kinds of people go, oh my God, we can probably figure out the genetic code. They thought it was going to take years, 25, 30, 50 years maybe, to figure out the genetic code. Probably less than 5 years they figured it all out. But here's the deal. Let's take our messenger RNA. Let's take a DNA. We're going to do a DNA. So let me write something up. Whenever a nucleic acid is copied, whether it's DNA or what, copied, the original strand is used as a template. And what do you make? The copy you make is going to be the complementary copy. It's going to be the complementary base, not the exact copy. It's a complement. I said this when we separated the two DNA strands and used each one as a template to make the original. The complement. So you had two originals that were, two copies that were identical to the original. So whenever a nucleic acid is copied, an original strand is used as a template, and a complement is made. And you have to use the Watson-Crick base pairing rules or the complementary base pairing rules. So let's just take DNA. Two strands. So here's our DNA. I'm going to put them far apart because I'm going to write something on them, okay? So, but I'm going to put several bases up here. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Okay, we'll just do that. I don't care how many I'm doing. Whatever. And let's just go ahead and blend it as people. Okay, so let's just put something up here. You want to give me some letters? A, G, T, C, A, C, G. No one's giving me any letters.
Yeah, you ran off before we could.
I was waiting for you. That's all.
Could it be like an AA?
Okay, A,
T.
T, G. All right, that's what you did. I don't really care what's up there.
So this is DNA. So
this is DNA. So yeah, so this is DNA. So this is one strand. One strand.
So the complementary is going to
we'll do the
be
complementary
T,
strand
C,
right now.
A, G, T, G, C, yeah.
Yeah, so once we know the one strand, we can immediately put it up. This is going to be
T, C, A, G, T, G,
G, C, A, T, A, C. And you asked if two A's can be together. Yeah, I could have A, C, 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 G, G. I mean, who knows what it is. It could be anything. So all I'm doing is just taking DNA, I'm kind of separating it so we can look at it. And I'm going to say one of those strands is going to be used to make messenger RNA. Notice that what is messenger RNA? A transcript or a copy of some gene. Now we have a tiny, tiny, tiny portion of a gene, but we're going to make a transcript of it. I don't care which copy size we use. Now I say I don't care which strand we use, but in reality, of course, enzymes know exactly which one to use because there are certain signals to tell this one or this one, whichever. They don't use both. They'll use one or the other. So let's just arbitrarily use this strand up here to make our, we'll say this is the strand we're going to use to make a messenger RNA transcript. I'm going to do it in different colors so you can see it. I'm going to do it red. So this is going to be my messenger RNA. And it's going to be a transcript of this guy. So let's just put these up. Okay. And you can tell me what the bases are going to be. So we're going to be copying this guy right here up above us. So what if there's an A up here? What do you? It's going to be a U. Can you see this? This is my red sign running out here. G is going to be a C. T is going to give us an A. C is going to give us a G. U. G. C. A, U, A, C.
So T.
I'm sorry, say it again.
A T transfers to A on in the RNA.
Yeah.
C, so T, yeah, no, T is still A. T and A still go together, but
When you have an
But-
A in
but when you
RNA,
have
there's
A,
only
when you make
uracil,
an RNA, you have to have a U.
no thymine.
So let me write this up. Now notice I'm going to explain some words here. So I'm going to rewrite this copy and rewrite my messenger RNA. Now that messenger RNA would be made in the nucleus, and then it's going to go out and it's going to hook up with some ribosome. So then here is our ribosome. Here is the messenger RNA hooking up with it. Well, let me just take this particular copy I just made. Let's see if I've ever read it. I'm losing it. I haven't read it here. Maybe this. That looks like not much better. Oh well. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So we have like U, C, A, G,
U, C, G,
C,
A, U, U,
A, U, U,
A, C.
A, C. Okay. Now, it so happens that, and this they actually figured out theoretically before they proved it for various things. If this is some kind of code sequence of bases to tell us how to make DNA, to make code for proteins, it can't be 1-to-1 because there's only 4 bases in the 20 amino acids. 2-to-1, that only gives a total of 16 combination. So it says it's got to be 3. It could be 4, but that's so many. So it's probably 3. And it turns out it is. And so that every set of 3 bases codes for 1 amino acid. And these are called codons. It's a codon. So every 3 bases of messenger RNA is a codon. That's the term, codon. And you see the word code in there because we're talking genetic code. So every 3 bases of messenger RNA is a codon, and each codon codes for 1 amino acid. Okay, each codon codes for 1 amino acid. So now the things I erased and all that, you can go back and see. I use the word codon, and I use the word anti-codon over there, and I'll talk about that in just a second. So yeah.
When you mean it codes, it gives the information.
It's the information. It's the information. And so of course this is what I'm talking about that they wanted to say. Once they realized this was a 3-base code. It's a 3-base code, meaning every 3 bases codes for 1 amino acid, and we call that each 3-base sequence we call it a codon. But they wanted to figure out what the code was, and the first one was discovered, and then suddenly, boy, everyone's trying to figure out the rest of the code, and it didn't take more than 5 years, and they figured out the entire code. And you'll see the code. It looks like. Actually, I didn't make enough copies for class.
I have one.
Just to hand it out, just so you can look at it. Otherwise, I don't have some. I'll show you. So it looks like this. And I'll make copies for it, we can use on Monday to talk about. But it looks like that. So the basic idea, without getting into details right now, is that each codon codes for 1 amino acid. But who is it that can recognize the codon and contains an amino acid? It's the transfer RNA. And so the transfer RNA has these little 3 bases down here that are called the anticodon, and they're going to pair by the regular base pairing rules. They're going to pair with the codon, and they carry an amino acid. So here's the basic idea, and I don't want to get any more complicated than this because it is too complicated. The basic idea is this. I'm going to erase some of this so you can see it. But this is the whole idea of protein synthesis. And, I think they probably have all kinds of animations you can get on YouTube and all that kind of stuff. I don't have this on. But the messenger RNA is going to start feeding through a ribosome. And so, on the messenger RNA are going to be these. Maybe I'm going to get bigger so we can see it. Their job here. Okay, so here's going to be my ribosome. It's got one small subunit, one big subunit, okay? And here comes the messenger RNA, and it's feeding through it like this. I'm going to put the arrow that way, saying it's going that direction. There are going to be 3 little bases here, 3 little bases here. Well, I should have been closer together here. Let's do it again. So here's a codon right here. Here's another codon. What's going to happen is those little transfer RNA guys. Now we tend to draw them this way when we try to make it easy. Instead of drawing out the fancy, we just tend to draw it like this. Here's the amino acid up here, and the anticodon's down here. That's a simple way of drawing a transfer RNA rather than drawing it up. So the first transfer RNA, the one that has the proper anticodon to recognize the first codon, got to be the right kind in order to fit. It's going to come down here like this and hook on together, temporary hooking on, and then here's amino acid number 1. Here comes another one that can hook onto this guy by whatever his anticodon is to match with this one. And we're going to say he's at amino acid number 2. I don't care what these amino acids are. This is number 1. This is number 2. There are enzymes involved that will then hook these together. And then the thing will shift by 1 codon. Just like those bottles shifting through the little pulleys, or whatever that system is where you're, or making cans of something. It's a factory. It shifts through. So suddenly it shifts through by 1 whole codon. And so now the codon's been shifted here like this. And these 3 guys, the 3 that were here are here. This guy gets removed. Okay. And what gets pushed into that place is this second guy. So now the second guy gets pushed over. So here's the second guy now, and he's going to look like this. He's still there. He's still hooked on to the second one. But what's he have on him? Amino acid number 2 hooked to amino acid number 1. And then a new transfer RNA comes in here with the new anticodon and a new amino acid number 3. Those guys, then the 2 gets hooked on to number 3 here. Shift another time, and another one comes down. It's a factory. You're not going to have to give me any of this information. That's not what I want. I want you to understand all the parts. But what I want you to realize is that it is like a factory. It is like a factory that the messenger RNA feeds through the ribosome.
So it's just like an
and as it goes through, the codons are read. They're read by the anticodons on the transfer RNAs. They hook on there. You get an enzymatic reaction to hook on this growing chain of amino acids on the next one. The chain gets longer and longer and longer. And when we look at the chart again on Monday, as we will, turns out there are punctuation marks. 3 of the codon sequences are said to be stop codons.
Yeah.
And when you get a stop codon in place that says we're done making the protein, release the protein and you're done. There's also a start sequence. So it's pretty wild, okay? So we'll kind of briefly do this on Monday, then we'll get into mitosis. And I'll tell you a little bit about mutations and everything. Hey, I still have 3 minutes to go. All right, see you guys on Monday.
Yep, see you.